everyone. Thanks for having me. It's fun to present my work to this community. I came to the meeting in Majorca many years ago, which is where actually this collaboration started with Christophe and Remy. But I haven't been back since, but I'm hoping to come again. And so what I'm going to present is experiments that I did. I was a postdoc at Woods Hole in biology and physical oceanography, working with Lauren Molyneux and Carl Helfrich, and these are experiments that we did with plankton and turbulence. And then eventually, using those observations, I did some more analysis with applying theories from Remy and Christophe, which took a long time, but it finally was just published. So here we go. We'll start with a little bit of background, and then there's two experimental biology papers that I'm going to be presenting. First, we're just going to think about how these plankton or larvae change their swimming in response to turbulence, and then focus more on their orientational response, and then interpret that in the context of this surfing theory. Okay. So one question that I'm interested in is just thinking about how plankton navigate in turbulence. One theme in our lab and in my research is thinking about transport. So I'm really interested in how things which are not fluid tracers are transported in a fluid flow. And we know plankton are small relative to the scales of turbulence, and that they can have both passive and active responses to the turbulence. And my question is, how do these responses together affect their net transport? Okay, so we know that when we consider plankton in the ocean, we know gravity is really important, right? There's both environmental and ecological stratification, so how things move up and down against gravity can control their environment. And also, many organisms are negatively buoyant, so they have to swim up to actively oppose gravity. So if you're a little plankton, you're swimming up against gravity, and say there's some turbulence, we know it can rotate you and make it harder to swim up against gravity. So many organisms have this displaced center of mass, so they're bottom heavy, so they can passively respond to help swim up even with under turbulence. And so there are a lot of studies of small organisms that have this passive response, or gyrotaxis, when we think of it in the viscous limit. And there's, this classic now science paper showing how gyrotactic plankton can get trapped due to the local shear in these thin layers. And so again, right, this is a passive response. So now there have been many studies studying these gyrotactic organisms in turbulence. So just to do a quick review, once you're swimming, you're no longer a fluid tracer, so you don't have to stay uniformly mixed in the fluid. And if you're a gyrotactic swimmer, you can form patches or preferentially accumulate in turbulence. In the shape, there have been some now many studies showing how the shape of the swimmer will control where it ends up in the turbulence. And there are a couple papers showing that if the swimmer is more spherical, it will preferentially sample downwelling areas of the turbulence. So that means they will not swim up as effectively in a turbulent flow, whereas if they're elongated, then they will actually preferentially sample upwelling areas of the flow, which could enhance their transport in some cases. And so there's a really nice figure illustrating this from this Levicchio et al. Paper in Science Advances, where they simulated gyrotactic swimmers. And on the left here is spherical swimmers, and on the right is elongated swimmers. And the color bar here shows the vertical velocity of the turbulent flow. And then here is a PDF of the vertical velocity of the flow that the swimmer is sampling. And you can see here that the spherical swimmers are observed in the downwelling flow more often, which would lead to reduced transport, and the elongated ones are more often in the upwelling flow, so they would be more likely to show enhanced transport. Okay, so okay, so there have been, so we know a little bit about gyrotactic swimmers in turbulence, and next, we're going to look at a really scientific plot I made here, which is studies in turbulence. All right, these gyrotactic phytoplankton are not the only plankton that people study in turbulence. But here is a number of studies in turbulence versus the Reynolds number of plankton based off their size and swim speed. And I'd argue that we have, relatively, there are there are some studies of phytoplankton in turbulence. We just saw some. And there are also a bunch of really interesting studies, too, of copepods in turbulence. But there are relatively fewer studies of invertebrate larvae in turbulence where they have this, who have an intermediate Reynolds number. So down here, we maybe are more in the Stokes flow regime. Up here, copepods can have their fast jumps where they can kind of escape the viscous limit. But down here in this more intermediate Reynolds number regime, there are relatively few studies. And part of it, too, is that the complex life cycle of invertebrate larvae makes it harder to do experiments with them. Okay, so the organism that I studied during for these studies is Crepidula fornicata, which is the Atlantic slipper snail. And we're specifically looking at its larvae. And so it has this shell and this upright posture with these velum, which are ciliated, and they beat their cilia to swim and feed. And they grow up to about 1 millimeter in diameter, and they can swim up to about, say, 4 millimeters per second. So if you estimate the Reynolds number, it's order 1, and here it's 4 in that example. And this is a video from Haoxi Zhang, who has these beautiful images of them, so where you can see them swimming. And what's nice is so they're bottom heavy, and they have this preference for swimming up, and they have rather smooth and variable swimming behaviors. So here is a video of them swimming in still water, and here are their trajectories being traced. And what you'll notice is they can swim side to side. They have some maybe helical motion, some exploratory motion. They don't have like very quick dive responses like some like oyster larvae or other sorts of plankton. Their behaviors are rather smooth and variable. So they can definitely, actively control their heading a little bit. So we found these to be a really useful model organism because they will be able to respond to the turbulence a little bit. And they're in this nice intermediate regime. Okay, so there have been some studies of larvae in turbulence. I'm just going to review a couple. But basically, here are some of sea urchin larvae. So they're not, they're maybe similar size, but, a different species. And they found that turbulence tended to tilt them over and made it so they did not swim up as well, and they tended to tilt with the turbulence. So they had, as you would expect, maybe a passive reorientational response. And then there's been some other models too that have applied this gyrotactic model to infer the larval orientation in turbulence. So kind of assuming the larvae are passively tilting. So this is orientation. There's also been a lot of really nice studies looking at how they modulate their swimming speed with respect to in turbulence. And so this is another paper led by Heidi Fuchs. And so this is a PNAS paper that was really nice. And they had these mud snail larvae. So they are similar. They're veligers. They're a little bit smaller than the organism we were looking at. And here they took them and they took two different species and they looked at their swimming response as a function of, say, dissipation rate, vorticity, and flow acceleration. And they looked at both. Here is like the normalized propulsive force, so basically how fast they're swimming, and whether or not they're sinking or diving, showing a dive response. And you can see that a lot of them are showing an increase in their swimmings with increasing, say, vorticity, dissipation rate, and acceleration. So we know that, so based off these studies, we might expect that bottom-heavy larvae will be tilted and maybe will have some passive gyrotactic response in turbulence, and they'll also swim faster under higher turbulence or higher turbulent cues. Okay, and then finally, so if they're responding to the turbulence, we need to think about how they can sense turbulence. And these veligers have statocysts, so these fluid-filled sacs that have this hard statolith. And they can use that to sense accelerations and rotations. So say they're accelerating, they can use this statocyst it will move. This relative motion can be used to interpret acceleration, and they can also be used theoretically to measure rotation. Okay, so that means we expect that they should be able to measure or sense some acceleration and rotation, could potentially respond to it either actively or passively or both. So yeah, we expect they're bottom heavy. And also, I didn't point out, but you saw the image of the villager. It's not very elongated, our little snail. It's, pretty spherical. So we'd expect that they would perhaps oversample downwelling areas of the flow, which would reduce their upward transport. But they also might increase their upward swimming in response to maybe balance that out. So that's what we went in thinking. So we did these laboratory experiments I did during my postdoc originally, where I worked at the Shore Lab in Woods Hole. And this was during the pandemic. So I don't know, I just like to show this picture. I had to have the snails in my basement to culture before I collected their larvae. And we ended up being able to run two sets of experiments with two different sets of the plankton. One I'll call the early stage. It was 2 days. They were 2 days old. They were smaller. They swam a little bit slower, 2 millimeters per second. And the late stage ones were older, 12 days old. They were bigger, and they could swim up to about 3 millimeters a second. And I didn't test whether or not they were competent. So whether they had reached competency and would be able to metamorphose, but we assume that they were at least approaching it. So they may have been more ready to settle and therefore have different behaviors. Okay, the experiments were in a jet-stirred turbulence tank. So we had this basically cubic tank, and there were jets in the corners. This is based off of Don Webster's tank, but it was slightly modified, and we can put a laser sheet with near IR laser to do measurements. There's a picture. I often give this talk to biologists, so I like to interdisciplinary audience, so I like to step through everything very carefully, but here's the tank. So we can image this center plane that we illuminate with the laser, and we can vary the turbulence level. It's, rather modest turbulence levels. And we put a bunch of larvae in the tank, and we run consecutive trials where we randomize the order of the turbulence level. So we keep changing the level of the turbulence and taking measurements. And in these 2D measurements, we get images like this where we can see particles in the back. There's many more. You can't see most of the particles in this image from the way the contrast is shown. But we can also see plankton, and we can track them when they intersect this 2D plane. So we do particle image velocimetry to get the flow field in the images. And we then also track the larvae in 2D as they move through the field of view. And so what are we actually measuring when we measure the larval velocity? We're interested in their behavior and their swimming, but there's a lot of things going on. So if we just think about a larva swimming in still water, we can approximate its dynamics, say, as some swimming velocity, some swim speed, and also some settling speed due to gravity. So the total relative velocity is just the sum of these two components, their swimming and some settling speed. Now, if they're in a turbulent flow, there's also the effects of the fluid velocity. So we can now approximate their total speed. So UP here, the plankton, is equal to this relative velocity plus the fluid velocity. And so what we're measuring is the total velocity UP. And then what we do is we interpolate the PIV vectors onto the center of the larva to estimate the fluid velocity. And then we subtract that to get the relative velocity, which is now a measure of the plankton swimming. So we're using the relative velocity as a proxy for swimming. We're not trying to estimate the swim speed directly by subtracting some settling velocity because there's so much uncertainty, or a little bit of, we didn't want to introduce that additional uncertainty. Okay, so here, let's look at what some of the data looks like. So first, just their behavior in still water. Oh, this got a little pixelated, I'm sorry. But this is our horizontal relative velocity and our vertical relative velocity. And here's just a PDF of the observations. So we see that they, swim, there's no bias in their horizontal swimming. The late stage larvae maybe swim a little bit faster. And in the vertical velocity, we see that the late stage larvae, so the older larvae, can swim both, swim faster up and also go faster downward. But the early stage larvae these younger larvae are more biased towards swimming up, and they show less variability in their swimming. But the maximum swim speed for these older larvae is a little bit higher than the young larvae. And I think, we had this earlier as well. Okay, so next what we're going to do is now we can measure, so this is what they look like in still water. Now we can observe them in turbulence. And what we're going to do is we're going to look at the upward swimming, so the upward relative velocity, or just the vertical relative velocity, and plot it as a function of the local flow acceleration. So we can measure that around the individuals. And so here we have the vertical swim speed, it's the relative velocity, against the local acceleration. And this is now all the observations together over all the levels of turbulence. And what we see is what we'd expect, or what we expected based off those, the other studies, that they swim up, they increase their upward swimming speed in response to the turbulence. And we see a stronger response for these early stage larvae than the late stage larvae. So we see that the larvae swim up in response to the flow accelerations of turbulence, and these younger larvae have a stronger response. Okay, so now what we were asking, well, this here, we've taken all of the observations from all the levels of turbulence, and we only looked at what was happening right around the individual. So we've inherently assumed that they, don't really have much memory. They're just responding to the flow around them. They're not integrating information over time. So instead we wanted to test, well, how important is, say, the background signal of the turbulence or their history? So are they responding to this instantaneous signal, or say the time average background noise, if you will? And here is just all our observations of acceleration, a PDF over the low, medium, and high turbulence level. And what we're asking is, so if you're in the middle here and you're feeling some intermediate acceleration event, does it matter will you respond the same way? If you are in a different level of background turbulence. So is the time average flow important? So here we have all our observations for reference. And so now what we can do is we can look at the same plot, but now I've separated by the turbulence level. So here's just the low turbulence level data, the medium, and the high. And what we see is that these plots are rather flat. So there's not much change in the local acceleration, but the biggest change is just between the different levels of turbulence. So what this indicates to us is that these early stage larvae are really integrating information in time and maybe not responding as strongly to the local flow around them. Whereas if we look at the late stage larvae, we see, some differences at the lower accelerations, but at the higher accelerations, all of the points have collapsed on top of each other, which means, if they are responding a bit to this, if they are responding, it's not till some critical value, and there's less of a difference. Their behavior is not as modulated by the background level of turbulence. And so we see that there are definitely differences in these two sets of individuals. And also the way that you interpret the data is really important, because here we would just naively expect that, or we would take away that they're responding to the local flow, but really it's maybe just the background level of turbulence here. So we see that they can, in some instances, integrate this turbulence information in time and respond to the background level of turbulence. And they are swimming up in response to, they're swimming up more in response to turbulence. They're increasing their swim speed. And this is similar to other villager larvae. So I've grabbed these plots without the axes labels, but it's their vertical swim speed going up in response to the local acceleration. Okay. Now I'm going to transition to talking about the orientation, but does anyone have a question at this point? No? Okay. So we see now how they swim up more in turbulence. They can respond to the turbulence, but what about their orientation? Okay, so they're biased towards swimming up. They're bottom heavy. We know turbulence has a lot of vorticity. And so if they're in some vorticity, they would the flow would tend to rotate them that way. They have some gravitational stabilization. So this is our passive reorientation model. And this is what we'd expect to see in our observation. So now we can just measure this, sort of. We can measure the vorticity around the plankton, and we can measure their relative horizontal direction as a proxy for their orientation. So now I have plotted the horizontal relative speed versus the vorticity around each individual. And then we binned all the data. And what we see is that when the plankton are in vorticity of a sign that would tend to tilt them to the left, we observe them swimming to the right. And when they're in vorticity that tend to tilt them to the right, we observe them swimming to the left. And we see this across all the levels of turbulence and in both stages of both sizes of the larvae. So this is the opposite of what we'd expect based off a model of passive reorientation. Okay, so this, we double, triple checked. It was like, do I have a sign difference? Nope, this was what the data looked like. So let's now think about what this means. So this is what we're observing when the larvae would be in vorticity tend to tilt them one way. We observe them swimming the opposite way. So this can't be explained by the torque due to gravity. So this must be some active orientational response, because they're pretty spherical, they're elongated, so they're really only responding or being rotated by the vorticity. And so here is an image. I spent a lot of time looking at the videos of the individual plankton to convince myself I didn't have a sign error in the data. And so here is one instance where here is its total velocity, here's its relative velocity, and here's the fluid velocity vectors relative to the plankton. So what we see is they're still bottom heavy, so they still have that passive stabilization in their upright position, but now they also have this active orientation behavior that you presumably would keep them upright in stronger turbulence. So that was kind of the initial takeaway, and I'll show you a video now of this image. So again, we have flow relative to the larva, the swimming, and the total larval velocity. And before I play it, right, these are 2D video. So the plankton kind of this plankton starts a little bit out of focus, and then it will come into focus and then go out. And hopefully it looks okay over Zoom. Now here it's out of focus, and it'll sort of come into the plane of focus, and you can see it's going to the right, and the flow is would tend to tilt it to the left. We can watch it again. I don't know how good it looks on Zoom. But yeah, so we're observing this sort of active rotational response. That opposes the local fluid vorticity. So now the question is, how does this relate to transport? Okay, so let's think about the vorticity. So here, the way I've drawn this in an XZ plane, my Z is my vertical coordinate. So this is negative omega, and so that would correspond to a positive dw dx. So that means the vertical velocity will be increasing to the right. So if this plankton is swimming to the right, it will tend to go towards more positive vertical velocity. So that means it would perhaps tend to sample more. I mean, you can see right here, if it goes to the right, it's going to be in flow that's going up. If it's, say, passively responding and going to the left, it would be in flow that's going more down. So that's why gyrotactic gives you often a preferential sampling of downwelling flow. In this case, it's going towards upwelling flow. And at the same time that I was looking at this data, this really awesome paper came out, Surfing on Turbulence, a strategy for planktonic navigation. And in this study, this was a theoretical study thinking, so say I have plankton and I assume that they can instantly reorient, they're memoryless, and they're only sensing the local velocity gradient. How can I say optimize their direction of swimming? So the only thing I change here is the orientation or the direction that they're swimming to maximize their total vertical velocity. And so, you know, if you just want to swim up, you might naively try to just swim up. But if you want to, say, take advantage of the turbulence, in some instances, you might want to tilt so you can surf the turbulence moving in the same direction as you. Okay, so in this strategy, they found that the surfing direction, so if you want to swim up, is to first order just up, then plus these higher order terms that are functions of the vertical velocity gradient. Here, modulated by tau, which is some time scale or some surfing horizon that you have to choose. And they found that, so this is just from this PRL paper. So here, all of the plankton were started at the Z naught and then left to go for a while. And then these circles here are the centroid after some time of all the points, and the passive ones have not really moved. The bottom heavy ones, so these are ones that just naively swim up constantly. So they're never reorienting. They're not even gyrotactic. They just swim up. They move up, but then the surfers swim up even more. So they can move up faster than the bottom heavy ones, even though they have the same swim speed. All they're doing is changing their orientation. And then here is a plot showing basically the effective velocity of the swimmers normalized by their swimming speed. So the effective velocity would have to do with their swimming plus the fluid flow. This is normalized by their swimming speed over the Kolmogorov speed. And we see that the surfers here, so the bottom heavy ones in this instance are always around 1 because that's on average, they're just going up at their swim speed. But the surfers are able to, in some instances, increase their swimming speed basically by 2 times. Increase it to twice their swim speed. Okay, so I was wondering, is this related at all to what we're observing in our experiments? So we started working together with Remy and Christoph, and Remy did a lot of nice simulations, and we were trying to compare our observations. So here now I've taken that same data before, but it's now normalized. So here is vorticity, now normalized by the Kolmogorov timescale, and here I have their horizontal relative velocity normalized by some estimate of their maximum swimming speed, right? In the model, they have a constant swim speed, but in our case, these are real animals. Their swim speed varies. And we see that the surfers here are doing the same thing as our plankton in that they're rotating to oppose the flow's local vorticity. Whereas the gyrotactic or passive responders tend to go with the vorticity, the surfers tend to oppose it. So this suggests that the plankton in our experiments might actually be able to surf the flow because they're surfing might not be the goal of their behavior, but it might be an outcome that comes from this active orientational response. Okay. So this is from the simulations of the surfers, and we see that they could be increased by up to, say, 2 times their swim speed. And that's due to the surfers preferentially sampling upward parts of the turbulence. So what we tried to do is see if we could measure any evidence of preferential sampling in our experiments. And so this is not trivial, but I'll walk you through it. So here I have the average dissipation for each trial. We have data points for the early and the late stage, the younger and the old larvae. And here what I've plotted is the average sampled vertical fluid velocity. So at all of my observations, I take the fluid velocity at the plankton, and I take the average, and then I take the measured fluid velocity places where the plankton is not, or just randomly, basically, throughout the flow to get an estimate of the mean vertical velocity in the tank. And I subtract it because there are small mean flows in the tank because it's a closed tank. But so that means here 0 means there's no preferential sampling. And this is what we get for all of our lowest levels of turbulence, which makes sense. The plankton are not preferentially sampling the turbulence. The turbulence is very strong. If we look at the medium and higher levels of turbulence, we see something slightly different. So if we look at the early stage larvae, they're kind of around 0. Maybe they start to negatively preferentially sample the flow at the highest turbulence, which means that we're actually observing them in downwelling parts of the flow more often. But we do get a lot of observations of the late stage larvae preferentially sampling upwelling parts of the flow. And so none of these plots are normal. This plot is not normalized. And that's because, right, we don't necessarily have one estimate of the swim speed, but on average, a lot of the larvae, they're swimming around 1 millimeter per second. And that's about what we're seeing. We're seeing that the preferential sampling is about at that order of magnitude, which is what we'd expect here. So this would tend to increase their net transport by about 2 times. Okay, so we see that there is perhaps some evidence of the late stage larvae here preferentially sampling upwelling flow, similar in magnitude to their swim speed. But we can't rule out that these early stage larvae are not preferentially sampling the flow. So these observations here of 0 and negative values don't necessarily rule it out. So we're in this closed tank, and the younger larvae, they can swim up really fast. So they're swimming up, they kind of get stuck near the top of the tank. So we're sort of biased by the ones that are stuck in the downwelling parts of the flow anyways. So what we need is a new experiment to actually measure this a little bit better. But what we're seeing is that there is this potential for enhancement to their transport. Okay. So maybe you believe me. So let's think more about the surfing theory. So the surfing theory, when it was introduced, let the plankton respond to the entire velocity gradient. But so far, we've just been talking about vorticity, which is just one part of the velocity gradient. And what Remy found is he found that when surfers use the full velocity gradient, they do tend to oppose the vorticity, which is what we found for our real plankton, but they also tend to align towards the maximal stretching direction in the rate of strain. So we haven't talked at all about the rate of strain in the flow yet. So we would expect our surfers to do this, both to rotate against the vorticity and to sort of align the rate of strain. So we decided to see if this is also going on in our experiments. So what we did is we estimated this angle just in 2D and looked at their orientation as a function of the rate of strain angle. And here's the rate of strain angle, and here's the relative horizontal velocity. And you can see it's rather flat. So we don't see any correlation with the strain angle. Our plankton are not biased one way or another, or at least the bias is not strong enough for us to pick up in the experiments. So our real plankton are not responding to the rate of strain in the same way that the true surfers are. They're only responding to the vorticity. So then what we decided, well, we've been comparing now to surfers that use the whole velocity gradient, but what if we now restrict the information that these theorized or simulated surfers get and only give them the fluid rotation? Can they still surf as well? Because that would help inform whether our real plankton are actually surfing. So then Remy went back and he simulated surfers with restricted information. So now this is the net velocity normalized by the wind speed. And this is a function of that time horizon. It's your basically surfing parameter. And it's typically optimized around 1 or 2 Kolmogorov timescales. And what you can see, so this is the net transport when given the full velocity gradient. And you can see that's the highest. The plankton have the best, surfing enhancement in that case. And then this green one here is when we only give them the rate of rotation tensor, and the blue here is when we only give them the rate of strain tensor. And while both information about the rate of strain tensor and the rate of rotation tensor seems to be important, if you only have to choose one, then responding to just the rate of rotation is going to get you a lot more velocity enhancement. We see it's much larger than just the strain. So we see that the rate of responding to the rate of rotation tensor is much more important than the rate of strain tensor, or responding to the vorticity is much more important. And one thing we haven't really talked about so much is, the Reynolds number. So Remy was able to do simulations at really low Reynolds number and then in high Reynolds number using the Johns Hopkins turbulence database. And he found similar results regardless of the Reynolds number, especially if you're around the optimal surfing. So that was good. And that means this is probably a robust result. Okay, so now let's summarize what I've gone over and ask the question, are the larvae surfing turbulence? So this is an AI photo. So one, we see that they do respond to the vorticity correctly on average. So our plankton are responding to the vorticity in the way we would expect these theoretical surfers to respond, so that's good. They aren't really responding, they are not responding to the fluid strain rate in the same way, but that's okay because responding to the strain rate doesn't get you as much in surfing. You can still surf pretty well, only responding to the vorticity. We do observe preferential sampling of the upwelling flow in some cases that could increase their transport relative to their swim speed. So that is good. And we also saw stronger preferential sampling in the late stage larvae, and they also had a stronger response to the vorticity. It was subtle, but and they did have a slightly stronger response, so that makes sense. But regardless, we did not see any evidence of them passively tilting with the turbulence and then having reduced transport due to that. So if anything, modeling these plankton as gyrotactic is not a good model. And yeah, effective surfing here we go. So it's just, to summarize the whole talk, we see that these plankton can integrate turbulence information over time. but also respond to the local turbulence signals around them. We see that they are actively stabilizing their orientation, and just assuming that they passively rotate may be an oversimplification, and they are potentially surfing. Okay, thanks, and I'm happy to take any questions.
Thank you very much. This is a great talk, and I love the drawing, by the way. Please feel free to ask questions. You can raise your hand or whatever.
What, can I start?
Yeah,
Yes,
hi,
sure.
Roman.
Okay. Michelle, thanks very much. Very interesting work. Two questions I know both might be difficult, and one maybe you've addressed a little bit. Why would they do that? That's maybe the easier one. And the second one is you sort of implied that they can measure any of this and just respond to whatever they want. How much is known there? Might there be indication that some of these cues, physical cues, are easier to measure and some of them not, or maybe we're completely in the dark?
So the first question is, why would they do this?
Yes.
Good question. So when these animals reproduce, they want to disperse far away from or disperse somewhere and find a suitable habitat. And staying up near the surface, especially in shallow waters, will help them be transported away and not feel the effects, the slower flow at the bottom. So that is, one idea at least, just to help them stay up in the water column. But it just could be a consequence of trying not to get turned upside down, that you just, they can also surf. The second question, do I have an idea of what is easiest to measure, what might be the strongest signal? I don't have a good answer to that. I know that they can also, I only mentioned here at the beginning the statocysts and how they use it they can measure acceleration and rotation. They also theoretically could measure strain with their deformation to the cilia, and that I didn't even mention. And I feel like that is probably the least strong signal relative to the acceleration and rotation, but I'm not sure of work that has really looked at, looked at how they no, sorry, I thought of a study, but I don't think it's relevant. I can't think of any work that answers that question off the top of my head, but it's very interesting and would be useful.
Thank you. Fascinating work.
Mm-hmm. Yeah.
Margaret.
Okay. That was great. I apologize if you addressed this already, but I have kind of an oddball question, which is response time, right? So like sensing is one thing, and that's a big puzzle. But once they've sensed the flow cue and then they have to sort of process it, like, are there any neuro people working on that? Because obviously, like the relevance timescales, that might be kind of interesting, thinking about turbulent timescales and then the response timescale between the plankton sensing the thing and then being able to respond to the thing. This is not a very well-posed question, but I was curious if you thought about it.
It's probably other people can answer that question better than me. I have not thought about that much. I know people think about this. I've been thinking about how we would measure it, and because it's a 3D system, right? I'm only taking 2D measurements. I had really wanted to try setting up 3D tracking, and maybe we'll be able to at some point, so we can just get longer trajectories and be able to actually see how they might, if there is some sort of temporal response we can measure. I know that of other species. I know people have been trying to look at this in other systems, but it's hard.
Like, could you do it with like a shear layer, right? Like if you set up like a really reliable shear layer and you saw them cross the boundary and that however long it took them to respond, I don't know if that would be like if you could make a facility like that, there'd be so many other things to think about. But yeah.
Yeah. I have mainly thought about it in the context of turbulence. Because right, you lose things when you make it more simplified, but also it's way harder to analyze. But I like the challenge. But I know maybe like Don has done some interesting work, right, where they have their animals responding to the shear layer. That gets at maybe some of what you're asking. Okay, lots of questions.
Yeah, before you get to the other questions, I was just going to interject. I think it's the Burgers vortex apparatus that you're thinking of. And Margaret, I think that'd be a really cool apparatus to look at how these animals are interacting with the details of a turbulent vortex. And who knows if there's latency. The copepods seem to react really almost instantaneously, but how the larvae would react is hard to know.
Yeah, I think you would be able to investigate that question really well with that facility, if I'm remembering correctly. So do things, more science.
Okay, anyone from IUI?
Can you hear me? Yes, hopefully. Thanks, Michelle. It was great, really interesting. I have a question. So in your maybe I'm getting it wrong, but in your plot of the the sensory response versus the transport up and down, it seems that if they track rotation only, then they'll have a negative response in when the turbulence picks up, right? So the green line goes below
Oh,
1.
so
Or
this
am I
here,
misinterpreting
yeah.
this?
So
Yes.
I didn't really explain this. So let me go back to the where I showed the surfing model. Okay, so in the surfing strategy, there is this basically free parameter tau, which is a time horizon, and in theory, the plankton choose it to be most optimal. So that is basic. Remy can kind of estimate a little bit. I guess there's a little bit of model to figure out what it should be, but also just would run simulations where he varied that and found the optimal value for each case. So if we go back to that plot, this one, or sure, this one. This is basically a control you would imagine the plankton has. So here is what they, if they were optimally surfing, they would choose it to be around 2, but we don't really know. This is sort of like a response time then.
Okay.
Does that make sense?
Yes, thank you. So this is the response time. Is there
Yeah.
anything okay, and you said already to Margaret that you don't know the response time at all.
I mean, because they seem to be doing similar behaviors to the optimal surfers, we assume it's probably optimal in this case, but it's not a true response time. It's just, it's an estimated, it's like how much they're weighting, the prior information.
Okay, very cool. Thank you very much.
Ricardo. Ricardo, yes, please go ahead if you have a question. We can't hear you.
Can you hear me now?
Yes.
Yes. Okay. Yeah, I had a problem with my. Sorry, it was a very interesting talk, and I was wondering whether the net speed that you were, or the swimming ability that you were measuring, whether it had anything to do with grazing strategy, and whether the larvae that you were using were in any way saturated with, they weren't trying to eat in any way or grazing or were.
So they were definitely eating all the little particles in there, and they're swimming and feeding current, right? Are related, or they swim and feed in a similar way. I can't really. I don't want to speak to whether or not this. I mean, maybe we did it with these two. These two sets of larvae. I would have loved to have done more experiments. I'm hoping to. I can't speak to whether this had to do with their grazing. I don't know. But that's a good question.
Yeah.
Mm-hmm. Great. Peter.
Yeah, thank you very much for the very nice talk. I got the tip from a colleague, Sören Amerkamp, and I'm a physical oceanographer, so not so much in the topic. But on one of your first slides, you showed the vertical velocity histogram in still water. And if I remember it correctly, it was up and down, right? So in both directions.
They go both up and down, yeah.
Yeah. And then I think 1 or 2 slides later, you showed the vertical velocity with a turbulence, and it was only upwards. Did I got it right? So
I only showed the mean in that case. Like it's the means bend.
So it still can be up and down, or what does it mean?
They still can be up and down. They definitely swim. The mean, though, is moving up in both cases as the turbulence is increased. And we see them swim up more under the turbulence in all cases. But they definitely, sometimes there are observations of them going down. Especially if they're in a really strong, I don't know, like every once in a while there will be one that's upside down because it probably got turned over in a strong event.
Okay, yeah. So
but there's a lot of variability, yeah.
So whenever there's a little bit of turbulence, the mean of all the phytoplankton goes upwards, right? I think that's amazing.
The mean of these plankton, go up. Yes. I'm sure there are probably scenarios I could construct where maybe it wouldn't, but I don't know if it was just like a tiny, barely imperceptible or, really, really strong. I think at some point there's some limit to the amount of turbulence that would make them happy. But in our experiments, we were in this, hopefully it was designed to be more relevant, low Reynolds number turbulence, not super, like in a breaking wave washing machine.
Okay, great. Thank you very much. Mm-hmm.
Okay, any more questions from?
I have a question.
Yes, please go ahead.
Actually, is there any ecological advantage of turbulence surfing for larvae, like the avoid predators, the energy saving, or the improvement in settlement?
So I know Remy did some analysis on the potential energy saving. I mean, if you, I would, if you want to go up and you're in turbulence, and if the turbulence would tend to, I guess you're waiting how you could go up faster by increasing your swim speed or by sort of tilting a little bit and trying to surf the turbulent eddies. And so you would have to compare the energetics of the tilting, the reorientation versus the increased propulsion. So, I mean, it would, in some cases, be energetically favorable to, surf these eddies if that was what you want to do, is go up. But that might.
I mean, with the turbulence, it can go up, or it can go up to the certain level for the settlement. I don't know. I'm just thinking in my head.
Yeah, I mean, these were not larvae trying to settle, because they were mostly swimming up in the experiment. How it would affect ones, say, that were competent, we gave them a settling cue. Not sure.
Okay, just one more question. Like, did the experiment with other zooplankton, like the copypod?
No,
I don't. Oh, okay.
no. There is some. I think one. I can't remember which study. There might be a copepod preferential sampling study, but it's hard. No one else has measured the same effect. So.
Okay. Well, so actually, in our lab, we work with the digital holography. So we image different types of plankton species, like the phytoplankton, the zooplankton. So that's why I was curious to know, did you try, or did you know there is any other literatures they did the same experiment with other species? So yeah.
I mean, there's not the exact same experiment. There are other observations of plankton and turbulence, but not ones that are looking for the surfing behavior yet.
Okay. Thanks.
Mm-hmm.
Okay, thank you very much, everyone. Thank you, Michelle, again for this wonderful talk. And I will just